the viral replication. The viral replication. And you know, like this viruses, if it will replicate, it disturb us, and if it will not replicate, it won't, you can say, disturb us. Okay, we need, you can say, in a certain quantity of viruses to, you can say, become ill. Okay, you can say, like a small number of viruses, they don't have the capacity to disturb us. We need, you can say, mass number of the viral particles that we refer as a virions to disturb our normal function, Now, the viral replication is the process by which you can say, by which virus increases their number. Okay, so it's the process, okay, by which viruses increasing, or you can say they increases their number. That's the meaning of viral replication. Now, so the viral replication is deal in the one-step growth curve. Okay, write it down. If we plot their fashion on the graph, it follows the one-step growth curve. It follows the one-step growth curve. So let's see how it is one step. So in the z-axis, we make one graph first. Here you can plot the number of virus 0, 10, 100, 1000, say 10,000. Okay, and here you can implement the time. Okay, here you can implement the time. So these two phases, what happened? The first virus will enter in our body. Okay, first viruses will enter in our body, and you know, whenever virus will enter, it suddenly not start, you can say, replication. Fine. It need the host, you can say, cellular environment. It have to first, you can say, established, you can say, very peaceful first relation with the host. In order to destroy, it first have to make themselves stable in the host environment, because hosts are also, you can say, designed in such a way that if some enemies come in their environment, they won't, you can say, welcome. Okay, they won't welcome. They must fight with their weapons, and their weapons is the immune cells. Okay, so first viruses have to make themselves stable, and that period is called as the eclipse period. Okay, that is referred as a eclipse period. So first virus enters, now they make themselves stable, and this period is called as the eclipse period. Now in this period, what is happening? What is happening here? First you plot the time, 0, 10, 20. It is in a minute. Okay, we have plotted the graph between the, you can say, number of progeny and the time. Okay, and the first thing is what? First is the exposure of virus in host. Okay, exposure of virus in the host, and then viruses have to make the attachment. Okay, viruses have to make the attachment, that is called as the initial attachment. In this phase, what is happening? Viruses will, you can say, attach with the host, and when they will, you can say, attached, then if first see with the diagram, suppose this is a host one cell, okay, and you know that we are the multicellular. If it is about the human as a host, then we have so many cells, okay, and suppose this is a respiratory, you can say the respiratory tissue, and these are the cells, and you know we have membrane bound genetic material or nucleus. Now what is happening, viruses, suppose these are the virus particles, it's entered. So the first incident is the attachment. So when it got attached, so if it attached with this is suppose 1 number cell, this is a 2 number cell, this is a 3 number cell, 4 and so on, okay. So if it attached with the first number of cell, okay, then this virus which you can say bind with the first number of cells or attached with the first number of cells, they loses the capacity to disturb the second or third or fourth. They are limited with only the first stage. Are you getting the point? Yeah. Once again, what is happening, the viruses enters, okay, and while they are entered, they make the attachment with the cells, and suppose if it is the first number cell, and if it got attached with the first number of the cell, then it will not affect the another cells, okay. First it will attach, and now it's a synthetic phase. Viruses, you can say, capture their requirement first, that's called the attachment, and now they will replicate in their acquired, you can say, nutrient site, their acquired cell site, okay, and that is called as the active synthesis. And this is about generally, if you talked about, then it is generally the 20 minutes. Only the 20 minutes it will apply. So now let's write it. The first step in the viral replication process is called as the eclipse period. Okay, it is called as the eclipse period. Fine. And in the eclipse period, what is happening? Initial attachment first, and this attachment will, you can say, disappear the viral's capacity to infect the other cell, okay. So you can say the pathogenicity of virus will disappear, okay, no problem. Now once they captured, now what they will do, they will multiply, okay. So that's called in this eclipse period, the another event after the attachment, losing, you can say, the capacity to infect the other cells, there is the active synthesis of what? Of their genetic material. Now the eclipse period is, you can say, followed by the exponential growth period. Exponential growth period. So the second event in the viral replication is the exponential growth. Remember, this eclipse period is about 20 minutes. It's about the 20 minutes. Now, the exponential growth is characterized by increase in the cellular number. See, from the, you can say, 10 virus enter, suppose, okay, and it will persistent, it will not go further because they loses the capacity. If 10 virus capture the 10 cell, and now there is active synthesis. Now these 10 cells, when they replicate enough, their, you can say, number increases. Their number goes, suppose it is about the 1000. Now their number are sufficient enough. Their number are sufficient enough to cause, you can say, disease, or to show the sign and symptoms in the host. So in the exponential growth, what is happening? The number of, you can say, progeny virus produced without, you can say, infected cells as increases exponentially for a period of time. Okay, and it is about the 8 to 70 minute. Write it down. It is about 8 to 70 minute. Okay, what is happening here? The number increases in exponential fashion, okay. So number increases in exponential phases, and the virus progeny number can be 100 to 1000 progeny. Got the points? Yeah. So once again, let's summarize what we studied. We studied about the viral replication, and we said that the viral replication is the process by which viruses increases their progeny, their number, okay. And this viral replication, you can say, is easily understood by the one-step growth curve, okay. The virus replication follows the one-step growth curve. So you can say, depicted in the y-axis, we depicted the number of virus progeny, and in the x-axis we presented the time period, okay. And we said that initially when the host got the viral exposure, then the viruses, if 10 virus will, you can say, came, if 10 virus is coming, then this 10 virus will capture the 10 cell, okay, and this 10 captured cell viruses loses the capacity to infect the other cells. They are only limited initially with the 10 cells, which means the viruses loses their capacity to cause infect the other cell, okay. The pathogenicity is temporarily disappear, fine. Now, and this period is called as the eclipse period. This period is called as the eclipse period, and it is about the 20 minutes, okay. And in this phase, the initial, you can say, the active synthesis, there is a active synthesis of their genome, their you can say genetic material. Now, this eclipse period is followed by the exponential growth, or you can say the exponential growth period. In the exponential growth period, what is happening? The number of virus progeny is increases in the exponential phase, okay, and it is about, you can say, 8 to 70 minutes. The virus number can be 100 to 1000. 100 to 1000. Understood? No problem? Yeah? So now, once we discussed about the viruses and viral replication, now let's move ahead and discuss that, fine, this is their, you can say, a mode of replication, okay, how they are replicating, how they are increasing their number. But fine, once they increase their number, they become potential enough to harm the host, to harm us. Now, how they are harming, in which way they are harming, okay, that's we discussed, and we already, you can say, make one conclusion that the viral, you can say, the infection manifestations we will see on the cellular level. So now we are going to discuss about our next topic, that's called the viral pathogenesis at cellular level.